These are on your disk. We just talked about it with several of the groups. What moved which way in the dialysis bag? Well, the question I was asked over here is what were the four things? Well, what were the four things that were moving? Water, IKI, starch, glucose. Keep in mind that materials move from areas of greater concentration to lesser concentration. But we had a restriction for that movement. It was called the dialysis bag. What was the restriction? What restriction was placed on those molecules and parts? Size. So the only ones that did not move were starch. The starch did not move. All the rest of them moved. How do we know they moved? Well, we know the water moved because the mass of the bag went up. So bringing that in, you need to know how to calculate percent mass. They give you the little formula for it in there. You will have to do this on the lab practical, so you better do some of those practice problems. All right? We know that the glucose moved. How do we know that? Benedict solution showed that there was glucose in the water in the beaker. We know that the starch didn't move because the outside did not turn. It stayed the same color as the iodine. We know that the iodine moved because the contents of the bag turned that blue black. Okay? Distinguish between the heated and non-heated yeast cells. Which ones absorb the red? The heated ones. Why? They were dead. What process could they not carry out then? Active transport. Both the heated and unheated, were carrying out what process? Passive transport, diffusion. Okay, identify an amoeba. Paramecium. That says Elodia. It should not say Elodia. It should say Euglena. Those were the 3 slides up here. You need to know, be able to look at it and say, oh, that's a Euglena. It is a protist. It is eukaryote. That's what you need to know. How do they move? That's this one. How does the amoeba move? What does it use?
That foot thing.
The foot thing, a pseudopod, yes. How do the paramecium move? Cilia. How do the Elodia, the Euglenas move? Flagella. What happens to plant cells when placed in a hypertonic solution? They shrivel up. What now can you see that you couldn't see before? What organelle or structure? Not the cell wall, but the cell membrane now becomes visible. You couldn't see it before because it was right up against the cell wall. What happens to an animal cell in a hypotonic solution? They swell up. What organelle did we see in the video that prevents it from bursting? Contractile vacuole, yes. If we put an animal cell in a hypertonic solution, it's going to shrivel also.
Is it going to die?
Yes.
Okay.
Especially if you don't get it back in water in a hurry. All right. The lab we did last week. You must know the parts of the microscope. I'm telling you right now, you're going to get to a station. There's going to be a microscope there. There's going to be 2 little Post-its like things on it. They're going to say, identify this structure. So they're going to pick 2 of them, and they're going to ask you to identify it. So you better look at it. The second bullet I have is describe the proper conditions for returning it. That's this. You better know them. How do you determine total magnification of the microscope?
Calculate the lens by the
Not subtract.
U times.
Yeah, you multiply the eyepiece or ocular times the objective lens. So if you have a 10 power ocular and a 40 power objective, the overall magnification is 400. Focus it under high power. I told you, you're going to have to do that. What's the relationship between focal distance and magnification? As the magnification increases, what happens to how far the lens is from the slide? It decreases. It gets closer and closer and closer. Determine the diameter of the field of view if you're given M1, M2, DFV1. That's this equation. M1 times DFV1 is equal to M2 times DFV2. Algebra. If I know 3 of those, I can figure out what the 4th one is. The likelihood is they're always going to ask you this one. What's the second field of view?
So the first field of view is always given?
Yeah, you've got to have one of them. You've got to have both magnifications. Keep in mind that because with a microscope you're measuring such small quantities, you can't measure in millimeters, which are small. You're going to measure in microns, which are, there's 1,000 of them per millimeter. You all know how to make a wet mount slide circle. You all know what happens if you're looking at an object and you push it away from you, it appears to come towards you. If you push it this way, it goes that way. Everything's opposite. Can't spell like C. Describe what happens to the diameter of the field of view if the magnification increases. Now, you look at a smaller and smaller area. Can you tell the difference between a plant cell and an animal cell? I hope so. And can you use a dichotomous T?
What's that?
That's where it says if you use this, then you go to this, if it's this, then you go to that.
Oh, that's the.
Like the bugs.
Okay.
Oh, yeah.
Okay? Okay, experiment 3. What's back? This is the one where we did the models, but we did them twice, if you recall. Back on the back shelf back there are the 4 molecules you're responsible for. You're also going to have to identify the functional groups by models. I'm telling you, as sure as you're there, you're going to get to a station and you're going to see a model of some kind in front of you, and they're going to ask you, is it a molecule or is it a functional group? If it's a molecule, which one is it? If it's a functional group, which one is it? Or maybe they're going to say, in this particular molecule, what functional groups do you see? That kind of thing, which was one of the questions that you had to do. You had to circle. You have to be able to do a dilution problem. How do you do that? What's the formula for it?
V1 times N1 equals V2 times N2.
Is it M?
I was thinking of
Volume times what? Concentration what?
I'm thinking of molar.
Yeah. V2, concentration 2. Again, same thing as this. If you're given 3 of them, you'll be able to figure it out. This is the one you're usually going to have to figure out. How much of that do you mix with water to make the dilution? Use a spectrophotometer. Those are spectrophotometers, folks. We have 2 calibrating knobs. What's this one calibrating? 0. How do you calibrate for 0? Well, you don't even know. You don't put anything in there. You don't put anything in for the 0. Then for the 100%, that's when you put the water in and set it with this knob, the other one. Those are the only 2 knobs you touch. Don't touch any other knobs. 0 without water. With water, with a clear solution, you set to 100%. Then you take out the water and put in whatever you want to get, and it's going to give you a percent transmittance. It will be less. Again, if you don't remember how to do this, folks, use your computers. Google it. You can find, I'm sure you can find a teaching one for it. Okay, use percent CT to determine concentration. Remember what we did? We made a graph that was kind of, I guess it was like this. And we had percent T concentration. I guess this was percent T concentration. The higher the concentration, the lower the percent. So we had that, and then we had an unknown. What did we do with the unknown? We measured the percent T, and we might find that it was right here. So we Draw a line right across here where it intersects the known line. Go down there and tell us if that's concentration. You need to know that. Go back into your lab and look at that. Okay, experiment 2. We've been using all of this stuff a lot. What were the 4 reagents we used in this experiment? You used 2 of them tonight. IKI and Benedict's. Biuret and Sudin 4. All right, Benedict's test for what? Tell me, you just did it. Glucose. What do you have to do with it? Eat it. All right, what test for starch? What test for protein? The biuret. But what do we have to add to the biuret?
Acid?
No, base. Sodium hydroxide. Sodium hydroxide.
I think that's the one when you're
Sudin
not
4
there.
tests for what? Lipids. How do we do it?
Acid?
No, we put it on a filter paper and soak it in the filter paper for a while, and then we wash it off and just look for a color change. Okay, second bullet. Why is the identification if we have an unknown? That could be starch, glucose, starch and glucose, or neither. Why do we always test for iodine first? I mean test for starch first.
Otherwise, it's a massive solution.
What now?
You don't need heat.
Yeah, and what happens to that reaction with heat? It interferes with the heat test. So you have to test with the iodine first, and then put Benedict's and test for glucose. That's the order of events. It has to be that way. You did that in the lab. They had you test it both ways and show you. Okay, in the acid hydrolysis experiment, that was the one where we took the starch from over here, we put some acid in it, we put it in a water bath, and then every minute we took a drop out and tested with iodine solution. Tested with iodine solution until there was no positive test, and then we tested it for glucose. But before we did that, since we had so much acid in it, we had to neutralize the acid. What did we add to it to neutralize the acid?
Sodium hydroxide.
We used sodium hydroxide again. And at the end of the hydrolysis experiment, we got a positive test for glucose. We had some pretty good results with that, as I recall. Okay, the very first lab you came in, first thing you did in this lab is you had a table and you were to make the metric conversions. And I had you circle certain ones, and that's pretty much what I have in parentheses here. You need to be able to also calculate surface area and volume of blocks, just like we did before, because you're going to be given a block or two, and you're going to be asked to calculate surface area and volume. Now, you only have a minute and a half. That won't be enough time for that. What I always tell my classes to do is get all of the measurements and write them on the back. You will eventually get to some stations where it'll take you nothing at all to answer because it'll be straightforward. And if you don't have enough time during the test itself to do those calculations, I will give you 10 or 15 minutes at the end of the test to work on the calculations. But at the end of the test, you are not allowed to go back and look at any other station. So it's your responsibility to get all of the information you need and write it on the back of the answer sheet that you'll have. Okay, what's the relationship between surface area and volume? As volume increases, what happens to the surface area proportionally? It decreases because we get that surface area to volume ratio. Why? Because surface area is a square of something. Volume is a cube of something. How do you determine volume by displacement?
You set it to, you take a graduated cylinder, you set it to a set point.
Yep.
You drop some in it, and you see the.
Yep. So you take one of these, put maybe 500 milliliters of water in it, and I drop an object in there, and it goes up to 600 milliliters of water. How much was the object?
100.
100. That's what you do. By the way, this is a graduated cylinder, in case you didn't know. How to use a balance? Well, you just used them again today. So that's why we did the things we did at the beginning of in our first lab, because you're using many of those techniques and instruments that we did then. Okay, what's a pipette? Well, you just used them over there. They're pretty accurate measurement devices. The last lab that you're going to do in here, we're going to use a micropipette that measures microliters. Fractions of a microliter.
Ooh, fun.
And levels of biological organization. In this lab, this was the one where you had atoms, molecules, organelles, cells, tissues. You walked around the room and looked at those different ones. You better review what the different